So, Stefan, you first joined the Max Perutz Labs for your PhD studies. At the time, the institute was founded. Now, over a decade later, you return as a group leader and professor. So, what's it like to be among the seniors now?
Obviously, time is flying, and I guess it makes me really realize that I'm getting old. But beyond that, I'm of course very happy about the opportunity to be able to return to the Max Perutz Labs in my new position as a professor of RNA biology, and I certainly have great memories from my time here as a PhD student. So I'm particularly excited about the possibility to provide to the younger generation the support and mentorship that I enjoyed in the past, also here at the Parrots. But particularly, I'm really looking forward to many scientific interactions with my colleagues, and I hope in the future we will be able to further contribute to the established status of Vienna as a hotspot for RNA research in Europe and beyond.
If you look back even further, when and why did you decide to become a researcher?
Well, I think that my interests in understanding molecular mechanisms in biology is somewhat rooted in my upbringing, because my father was an entrepreneur in the engineering field where I frequently helped out to put machines together. And my mother was working as a nurse in a children's hospital. So I guess that this somehow set the basis and the boundaries for my interests in later on systematically trying to dissect the molecular machines, and that in a biomedical research settings. But of course, on the way there, there were many influential teachers, lecturers, mentors, and colleagues that had an immense impact on my passion for science.
I noticed the guitar in your office, so I really wondered if this was a career you have considered.
Well, early on, sure, but then recognizing my limitations made me choose wisely on the future career choice. But it's certainly still a hobby that I have. And looking back onto my PhD times, I think it was a great time to assemble a band of PhD students and even play on the roof of the Perutz Labs. So these are great memories, and maybe who knows, there will be the chance in future to assemble a couple of colleagues to revive this. Who knows?
So let's talk about your research now. You did your PhD in the lab of René Schröder, an expert in RNA biology, and you yourself are now focused on the role of RNA modifications in the regulation of RNA fate and function. What fascinates you about RNA?
RNA, in one form or another, impacts really every process in a cell. And this is really possible because RNA carries out a broad range of functions, from transmitting the genetic information into molecular machines and structural building blocks of the cell, to the regulation of gene expression during development, cellular differentiation, and in reaction to the changing environment. And these broad functions are really rooted in the fact that life on Earth may have really begun with a simple RNA molecule. And this is what the RNA world hypothesis suggests, because RNA combines really the ability to both store genetic information like DNA does now, and drive chemical reactions like enzymes do now. And many of these versatile functions are still found in current biological systems. And in that way, one can really refer to RNA as the central molecule in life. And as such, it has surprised many generations of scientists and will certainly continue to do so in future.
What do you think are the big open questions in your field?
In contrast to what is frequently portrayed as a static picture that many assays provide when inspecting gene expression, this process is actually involving a highly dynamic interplay of many steps from RNA synthesis to decay. So I think that one of the major challenges is really to develop tools that allow us to monitor and measure such highly dynamic processes inside living cells. And what we do is we really try to develop and employ novel technologies such as SLAM-seq that allow us to produce molecular movies of gene expression rather than static images, with a goal to capture and quantitate such dynamic processes. And with such assays in hand, we are really trying to uncover and dissect Novel and fundamental biological processes that control gene expression.
You mentioned education. Thinking of yourself when you did your PhD here at the Perutz, with the experience of 8+ years as a group leader, what would you do differently? What is your advice to students?
Honestly, I'm not sure I would do many things entirely different, which certainly does not mean that I have made no mistakes. I certainly made many of those, but I feel I have learned a lot from those mistakes. So my advice to students would be to be really courageous and not fearful of what the future may bring. I think now is probably one of the best times to do science with endless possibilities. And while I do acknowledge that the competition is very high, particularly in the academic setting, I would say that as long as you do what you are really passionate about and what truly fascinates you, there will most certainly be a path forward.